So, you ready to start filtering?
Yeah, I'm excited to see this.
Me too. I was so relieved that the pump actually powered on when we needed
phew.
to. Total few. It's always a nervous moment, but it hasn't let me down yet. So now what we're going to do, I'm going to do my job of getting this water through that filter into this Erlenmeyer and giving you a volume. I'm going to ask you for some start times and end times in just a sec. So I'm just I'm going to just start up Greenburn Lake Site C, and can you give me the start time for that, please?
Yeah, so Greenburn Lake Site C is 1801.
Perfect, thank you. I'll just add some water into here and we're good to go. So that's rolling, and I've written...
So Jared, why do you write it on the top there?
Well, I don't want to keep bugging you, so I want to keep track because I know that in 25 minutes that filter is going to clog. And I don't want to keep asking you how long's it been. So if I write that down there, then I don't have to bother you again because I know you're busy with data entry. So let's get going on the next one too. And we're actually crossing the site barrier here. I'm going to start entering Beaver Pond 1A. And so can you give me the start time for Beaver Pond 1A?
Yeah, Beaver Pond 1A is also 601.
Good, I loved your paraphrasing there. That was really nice. So that's perfect. I'll just write that on top. And now we're filtering away. So 601, and it's Beaver Pond 1A. But got it. Awesome. And I know we use military times, but we're fine to say 601 between us. Okay. But military times are going in the data. I see that. That's awesome. Okay. So in theory, we just have to let the water draw through here. I noticed that the Beaver Pond one, and this is kind of neat, Ray. Did you notice that yesterday when we filled Beaver Pond, or sorry, this morning, that Beaver Pond one was the one that with all that scudge on top, and it looked like just a terrible sight. But yet it's filtering a lot faster. And even if you look at the filter paper, you can see this is whiter than that. This is getting dirty already.
That's from the tannins,
right? It's
the
tannins. That's exactly what I was going to say. And so Greenburn Lake, although the water actually looked clearer when we did the top-down check, it actually looked pretty good compared to this one. Beaver Pond just had a lot of suspended particulate matter, and that doesn't actually slow down the filter. They're fine. But when there's tannins, even though it looked clear, there was some yellowish tinge, that really slows down filtration. And you can now see those tannins being caught on that filter. It's much dirtier. And so it's not always what you see is what you get. Sometimes there's some surprises, and tannins are the devil. So
you probably have to account for that when you go out sampling if you're doing a lot of heavy tannin lakes.
You do. Especially it becomes problematic up north where there's a lot of muskeg, and becomes problematic on the east coast. So what I'm doing here is just as that filter runs dry, I just keep topping it up continually. And my objective there is to get as much water through. There's no prescribed volume. I just want as much as possible to get through that filter. The more water I get through, the greater likelihood that I will capture that single strand of eDNA that happens to be in the bottle. Maybe there's 10, maybe there's 1, we don't know. And so I want as much water through as possible. So this takes generally about 25 minutes per replicate. What happens is in that after about 25 minutes, and this is a really hard point to explain to people, but experience has shown me that with a 0.45 millimeter or nanometer pore size, the filter typically will clog in about 25 to 30 minutes when you're running a pump at about 20 psi. Now, the volume of water you've got through in that time will vary widely, depending if it was a Lodic site, nice clear stream water. See how fast Beaver Pond's going. Or if it's a really crappy sample from a lake with a lot of tannins, that volume might just be 300 mils through a really tannin-infected lake.
You don't have to filter for an hour just to get the full amount of water.
You won't. After 25 minutes, it's going to stop.
Okay.
You won't, as much as you want, it won't go through anymore. The filter's clogged.
So just end a sample at 25 minutes.
Well, technically, you want to watch for when there's no more drops coming down that tube. And when that's indicating there's no more drops coming down, that tells you the filter is clogged and you're done. There's nothing more you can do. You can keep the thing running all night. You won't get any more water through because it is clogged. It's like a plug in the sink.
Yeah.
And so basically the goal is to just get as much water as you can through before it plugs, which is why it's so important that we ensure our samples are clean. You remember we checked down in the field? That was to make sure we didn't have a ton of muck in here. And if we can do better, we should do better. Because the better site water we get, the more I'm going to get through the filter, the greater probability I'm going to get DNA on my
It makes
sample.
our job easier in the lab.
A lot easier.
Yeah.
But if you go to like a Lodic site with some super clear tailed frog, really good water, you would drink it right out of the stream kind of site, you might get 2 or 3 liters through. So when I see really clear water, I sometimes collect more than a liter. I sometimes might collect a 3-liter sample for A and 3 liters for B and 3 liters for C. And you'll be like, oh my gosh, I'll spend all night filtering, but no, you'll find it goes through incredibly quickly.
Okay.
And so to give you a contrast, and a stark contrast, if you filter distilled water, which we'll do with our field blank at the end of the day, distilled water at 20 psi will go through that filter in 2 minutes.
Wow, that's incredible.
So lake water will go through, we'll get about 900 mils, 600 mils through in 25 minutes. Stream water, I'll get a liter through in about 8 to 10 minutes. And so knowing that, you can kind of predict your sample times. So the best case scenario, the best you can ever do is distilled water, 2 minutes, it's done, whole liter. The next best is stream water, nice and clear. You probably get 1, 2, 3 liters through. I still, most of the time, just do 1, it's good enough. But the worst you can do is lakes. That's the hardest. They're tough to sample, so just be prepared for that. We can get some great results, but it does go slower. So I'm just going to basically keep topping this up until I've got as much water as I can through, which I'll know when that stops flowing, when I stop seeing water through there.
Cool, so I guess we just wait.
Yeah, so now we just wait till that's all dry, and once they're dry, we let them, once we've got either all the water through, we're just out of water, or the filter was so clogged that we can't get any more, we've given up, we're just going to let it pull air through for about 3 to 5 minutes. And that's just getting the final bits of water, getting as dry as we can before we put it in the envelope. And then we let the silica do the rest of the work. It'll do the last 5%. But I want to give it the driest filter paper I can.
Yeah, that makes sense.
Perfect. So that's good. It's just a waiting game. Well, this is great. We actually got through all of our water from Beaver Pond 1, got through 1,000 mil. From the Greenburn Lake sample with more tannins, we only got through about 700 mil.
Holy, and how long has it been?
They both took about 25 minutes, and then they clogged up. And so it's pretty much exactly as I said. And you can see if you look at the filter, there's a lot more tannins on that one too. So yeah. So now we're ready to do the next step in the process. I've let this run for 3 to 5 minutes. So we've taken off as much water as I can using the pump. So at this point, I can kill the pump, which is a blessed relief for the sound.
I recorded the end times.
Yep, you gave me the end time. What was it again?
The end
for
time.
For Greenburn 1?
Yeah, Greenburn 1 was 1826.
Perfect. So that's 25 minutes on that one. That's perfect. And then for the BP01A, it was also the same. What was that?
It was also 1826.
So perfect. So they both logged up at about 25 minutes, but fortunately I got all of the BP1 through Beaver Pond, but only some of the Greenburn. Okay. So that's terrific. So now we can put these bottles away. These are clearly labeled. And thanks for that nice chain of custody and repetition too. That's good. So the last thing I need to give you is
the volume.
Perfect. I'm going to give you the volume for Greenburn 1A right now.
Okay.
So I just disconnect things. I pour it into here. I measure it up. And it turns out it's 700 mil.
So the volume for Greenburn 1A is 700 mils.
Perfect. I'm just going to check that behind. And then now all I have to do is separate this from this. We're kind of done with this equipment now. I've just got this to go through the filtering. I'm just going to give you the volume for Beaver Pond 1A now. So, and the volume for Beaver Pond 1A was 1,000 mil.
Okay, so the volume for Beaver Pond A is 1,000 mils.
Beaver Pond 1A.
1A is 1,000 mils.
So again, I just removed this, and there's no screw or thread. It's just, you just sort of pull it off gently.
You have to rock it off, right? Because it's all threaded?
Well, the real rocking actually comes here, because there's no threading here. It's not too hard to separate off our equipment here. So just to make a little bit of space, and we're done filtering for the night. We've got all our samples through. I'm just going to give myself a little bit more room. And I'm going to move my bleach, 50-50 bleach and water, my distilled water rinse number 1, and my distilled water rinse number 2 up here. Now my task is simple. I'll put Beaver Pond aside for now. I have to get the filter paper off here and into that envelope. You've labeled my envelopes. You've got the data entered in Excel, so we're ready to go there. My hands are dirty because I've been touching bottles and all sorts of things. So at this point, I could wear gloves if I want to, but I'm going to just be really, really careful with the forceps. Good lab practices would dictate that you do wear gloves, but I find I lose some dexterity, and I trust myself enough to know that I will never touch inside this filter funnel, and I will never touch the filter paper. So let's go ahead and just do that. So the first step is just to separate this, and it takes a bit of force, but it pops off, and I put the cap on it so I still know which sample it is. I'm
So
fine
the wind
that way.
doesn't take it away.
And this goes into our recycling area so that we're fine. Yeah, and it basically just protects it if I happen to drop water from another bottle over it while I'm doing this, like a little drop falls off or something if we're still filtering, then that just gives it a bit of protection. So now it's ready to go. I'm just going to clear my forceps. So I'm trying to get any residual excess bleach off those forceps. Give them a little rinse, get rid of that water, a little rinse, and now a good tap to get rid of that water, because any water I introduce is just letting those exonucleases go to work again. And we've spent a lot of time trying to get them off this filter. So now I'll just expose that, and I very carefully, without talking over, it doesn't matter so much because they're all tested against human DNA, but without certainly touching that, I just fold that in half and I crimp the edge, and that confuses a lot of people. I'm using the cardboard backing at the back to turn my stuff around when I need to turn it. I'm not doing it using the filter paper because it'll tear. This is very soft. It's like wet toilet paper, and so you got to be gentle with that. And with that, I've now got my filter paper ready. The one other thing I want to explain is this is the filter. This is the filter backing. I have had people accidentally send the backing to the lab and throw away the filter. So don't do that. We want to make sure you're sending the filter paper through. So if you can open the envelope for me, that would be great. And I'm going to put the filter paper right in.
I have it open here, but I understand that if you can't get it open, you don't want to stick your finger in.
You do not.
But you can blow in it.
Yeah, a sharp, quick blow into it will pop the envelope open. You can crimp the side, and that holds it.
But you have to make sure you don't have your target taxa for lunch.
Yes, if you've eaten a sockeye salmon sandwich and then you're testing for sockeye and you blew in that would definitely get a hit. But if you've not eaten a red leg frog, we're okay. You can blow in that.
So many possible sources of contamination.
I know, that's why I have to be so careful. The other little trick that sometimes happens, I was really careful to watch to make sure that filter paper did drop inside your coin envelope. I've seen it happen before where it was stuck to the tweezers and the person didn't look and they brought it back out and it fell straight into the bleach cup. And it was just a bad coincidence, but that was done. So that's great. So now I'm done with this. You're doing that. You can lick that or seal it any way you want. You can use your sponge with water, or in our case, we now have sticky ones. I'm going to put these back in bleach. Now, if I'm doing the same site, theoretically, I wouldn't have to put them back in bleach if it's all Greenburn Lake. But I don't want to get into bad habits because in this case, we're actually crossing the site barrier now. We're going to another site, a Lodex site. And so I just make it a good lab practice to always bleach my forceps between each sample.
So maybe we'll get this one packed up before we start that site?
That sounds good. So why don't you go ahead and add silica desiccant? And in theory, I know we're just doing a video here in the interest of time. We've just got one coin envelope, but in theory, there would be 3 coin envelopes. And that's enough. That's plenty. So yeah. So there'd be 3 coin envelopes in a bag. That Ziploc bag gets labeled with a Sharpie, which we're not going to do in the interest of time. And you compress the air out of it, and you seal it up, and that can go to the lab. And that's our complete sample. So for the purpose of demonstration, everyone's going to get, there you go. It's done. In the fridge or in the freezer if we've got one is the best. So, and we'll courier that off to the lab in the next day or so when we finish collection. We have a minute. So.
Great.
Cool. So I'm going to do that one more time with the site from Beaver Pond. So Reagan, I'm just going to actually take the filter. We're going to repeat the same thing we just did for Greenburn Lake, and we're going to do it for Beaver Pond now, for Beaver Pond 1A. So I just need to separate, whoops, I'll keep that on, separate this. It takes a bit of controlled force. Jeepers, these bottles are really hard, or these filters. Mine come off a little easier. Okay, so there we go. Cover that just to protect it. And now this dirty stuff is all the filtride, the DNA, that I'm hoping sent to the lab. I don't worry if there's little bits of duckweed and stuff stuck there. The lab's not going to be bothered by that. They're just going to cut it into quarters and then process it. So that's all fine. And if we're working around this or moving other bottles over it, I make sure I have the lid back on. But that's basically the filter paper. That's my bleach, water, water. So I'm going to go ahead and remove that. So good double tap, get the bleach off. Double rinse. Technically, I should actually have this over while I'm doing this because there's potential for water drops, and a good double rinse here. And again, I'm not wearing gloves because I'm going to just follow really good practices and not touch things. So I cut it in half, or fold it in half, crease the seam. This is about as soft as toilet paper, wet toilet paper now, so I'm gentle to crease it with the flat side of the tweezers, not the sharp side, or it'll just tear. Then I fold it in half again, so it's now folded into quarters. The lab will use each quarter to test for their focal taxa. Then I just take that up. I'm not bringing the cardboard. I don't care about that. That's now garbage. I put it in the coin envelope, and I make sure it drops right into the bottom of the coin envelope. So that's fantastic. And then it's up to you to go ahead and seal that. So you can just use the sticky labels. If you had the lick-on glue adhesives, then you would just basically lick them, and that's okay. So we're good. So that's perfect. I can go to the lab now, and we're good to go. We'll wait for our exciting results.
Boom, boom, boom.
Cool.
Nice.